Preparations of temporary slides. Making microscopic specimens for viewing usually involves cutting thin slices of the specimen, especially of plant specimen. The cut slices are known as sections. There are two ways of making sections. The first one is by cutting or slicing the specimen longitudinally, that is, along the length of the specimen. For example, if this is a stem, if the cutting is done along the length, that is the plane of cutting, then there will be two halves produced that display the longitudinal section. So when this stem is cut into half longitudinally, what would be obtained are these longitudinal sections, and they display the internal tissues along the length of the stem. The second way of making the section is by cutting the specimen transversely, that is, across the length. If this is a stem, if the sectioning is done across the length like that, this is the plane of cutting, then what would be displayed would be a cross section. So this is the cross section of the stem. You note that the display of tissues in a cross section is very different from the display of tissues in a longitudinal section. They're the same tissues, but the way they're displayed are quite different. So it is very important to know the sections of the specimen that have been displayed, because each has a unique way of displaying the sections. Now, when preparing the temporary slides, there are certain precautions that must be taken, and these precautions include: 1. The sections must be very thin. They must be very thin. Why? This will allow light rays to pass through the specimen so as to reach the lenses for illumination and magnification. Remember, in a light microscope, the image of the specimen being observed is made when light rays pass through the specimen. So it is very important that the specimen be as thin as possible to allow the light rays to pass through. The second precaution is that the scalpel or the razor blade that is used to cut the sections must be very sharp. This enables making of not only thin sections but smooth ones, and in this way distortions of the cells is prevented, because if the scalpel is blunt, then cells are going to be distorted, because you're not going to have a clean cut of the section, but instead some of the cells and tissues will be pushed out of place, and that will distort the appearance of the tissues. The third precaution is that the sections on the slide must be kept wet. This is done by placing the section in a drop of water on the microscope slide. The water prevents the tissues from drying, which would otherwise distort the cells. In addition, turgid cells are larger and thus easier to view. Now, sometimes it is necessary to stain the specimen under observation. This is because in most cells the structures are colorless. In fact, most living cells are colorless and transparent. Thus, they're not so easy to view. For this reason, different types of biological stains. There are different types of biological stains that are used. These stains include iodine solution and methylene blue. These are the stains that are commonly used in the laboratory, and in particular when working with plant specimens. Most of the stains, including these two, are selectively picked up by the plant tissue so that they selectively color different parts or constituents of the cell or tissue, thus creating contrast for clearer viewing. In this particular case, this is a cross-section of a stem. Without the staining, then the tissues appear as transparent, and it's not so easy to distinguish between one and the other. But with staining, some of the tissues pick up the stain, and so they stand out in clear contrast compared to the others. So this makes the viewing very easy, and it will be easy to identify and therefore study specific tissues and observation.